Okay, well, I think we've had a few more people show up, and I'm sure, people will roll in as they're able. So I'll go ahead and get started with the land acknowledgement like we normally start these seminars. So we respectfully acknowledge the territory in which Memorial's campuses are located as the ancestral homelands of the Beothuk and the island of Newfoundland as the ancestral homelands of the Mi'kmaq and Beothuk. We would also like to recognize the Inuit of Nunatsiavut and Nunatukavut and the Inuit Nitassinan and their ancestors as the original people of Labrador. We strive for respectful relationships with all the people of this province as we search for collective healing and true reconciliation and honor this beautiful land together. So on that note, we are ready to start. Our speaker this week, Friedrich, you can start whenever you're ready.
Yes. All right. Thank you very much. Thanks so much for inviting me. It's an honor to be here. I'm just gonna share this, and hopefully you can see that. All right. My name is Fredrik Jutfeldt, and I will be talking about fish, as you can see. So the topic is thermal acclimation and adaptation. I will talk about two different experiments, one on acclimation and one on adaptation. First of all, I want to say where I'm from. So this is my university, NTNU, in Trondheim in Norway. And this is pretty much what it looks like right now. It's very snowy, quite cold. I imagine climate is similar to what you guys have. This is where I do my marine work mostly. This is Kristineberg Marine Station in Sweden. And it's a beautiful place for marine research. The station is these buildings in the front there with good access to the water. But I actually do mostly zebrafish research during most of the year, and then do some trips to do marine work. So the research in my lab, we do ecophysiology and environmental with the focus on physiology. So for example, then the mechanisms of thermal tolerance and thermal performance, what kind of physiological functions determine these things. Thermal acclimation capacity and adaptation, evolutionary adaptation. So those are the two things I'll talk about today. And also we in my lab, we do ocean acidification work on fish physiology and behavior. And over the last years, I've also been involved with a bit of meta-science work and scientific misconduct, looking into data fabrication and things like that. But I won't go into that right now. So first of all, I need to acknowledge the people who actually did the work that I'm going to present. So if I happen to say we, I mean, or if I happen to say I mean we, and if I say we, I mean they, because I just take credit for other people's work mostly. So these are the people who actually did most of the work. All right, I'm not gonna dwell on climate change, but we know we have very varying projections from quite minor changes in climate to huge climate warming, depending on what we do. So we know that the average temperatures go up, and we know that extreme temperatures can be more common. For example, heat waves. So for average temperatures, the thinking is that thermal performance or long-term performance of animal function can be affected in different ways, while these heat waves over a shorter time may be more relevant for the thermal tolerance, the acute thermal tolerance of the organisms. And animals have various ways of coping with this. They migrate, we know this. This is well mapped. They can acclimate, individuals can acclimate, and populations can adapt through evolution. And in my lab, we're looking at these two last functions there, acclimation and adaptation, and those are the two I will focus on today. Coming from a marine, mostly working on marine organisms, I switched to zebrafish for quite good reasons. I think it's an extremely practical organism, and that is why it's used in a lot of biomedical research, being one of the most common experimental animals right now. And it is extremely practical. It's very hardy and fast growing. It also, for my research, it covers quite a wide thermal range, and it can acclimate rapidly to various temperatures, and that makes it convenient for my questions. It grows fast, so we can run growth experiments over weeks instead of months. It has a short generation time, and that's really the key, because that's really the main reason I picked zebrafish, because it allows us to do multi-generation work, which is very difficult in marine fishes. Before I started studying zebrafish properly, we went to India to investigate a bit what they do in India. And here's a little video of where we found zebrafish in India. So they are in very shallow water, right next to the yeah, you can see this is in a sandy pond. Here they are right along the seagrass, along the edges of the river in very shallow water. And they're not afraid at all. They come right up to the camera, and they are quite easy to catch. So it's not strange that they are a good model animal, because they live in such small spaces and in dense populations. So they were probably easy to domesticate. So the wild fish in India, they live in these ponds among the plant, and then the lab fish. So these zebrafish were domesticated in the 70s, and they have been in biomedical research facilities for the last 150 generations, roughly. So that has really domesticated the fish and made them good, function well in small tanks and eating artificial food and all of that. But we also know that the water temperatures in the wild can fluctuate dramatically over seasons. So in the wet season, there's a lot of water coming down from the Himalayas in northern India, and that brings down cold water. And then in the dry season, it can become stagnant and shallow, slow-moving rivers and very hot water temperatures. So we think that they have a good capacity to acclimate to these different temperatures. While in the lab, the fish have been at 28 degrees for 150 generations with almost no thermal fluctuation. So we wanted to dig into that, what has happened to the fish a little bit more. But before we talk about that, we need to talk about plasticity. So plasticity is something ecologists use a lot for describing how organisms change with environmental change. So a trait, you can measure a trait, and then if the environment changes, a highly plastic organism can have a high response, a trait change, and a low responding organism can have a lower trait change over that environmental gradient. But for us animal physiologists working on temperature, it can be a little bit different, because acclimation capacity is a form of plasticity. It's something that the organism does as a response to temperature change. But they do it to compensate for thermal forcing. So if we increase the temperature, we know that the biological rates of an organism, the metabolic rate or enzymatic rates, increase with temperature. And if the animal doesn't do anything, it actually has a steep trait change. While after acclimation, animals that acclimate well have a lower rate change or a lower or shallower response to this environmental change. So It's actually the opposite here, in that the ones that I would call say to have low plasticity, they actually have a steeper gradient, while those with a high plasticity can maintain normal function over a bigger, larger temperature range. So they're less affected by temperature. And this is important for seasonal acclimation, for example. Ecologists also ask what the cost is of plasticity, because why aren't organisms more plastic? Why aren't they infinitely plastic? Why can't they just adjust their biology, physiology, and biochemistry to any environmental change, really? So we wanted to dig into that cost of plasticity. So we came up with a hypothesis that fish in the wild, they are adapted to large thermal fluctuations over seasons. And these lab zebrafish from the same species, but isolated in the lab for 150 generations, they are adapted to stable and optimal temperatures. So we can predict how these two different populations would respond to temperature. So if we allow them acclimation to different temperatures here, so these are acclimation temperatures, we predict that the performance of the lab fish in blue here have a sharp peak around 28 degrees, where they have optimal performance. But then if temperature changes outside of that, the performance deteriorates faster, and that the wild fish have in green here, have a broader thermal breadth. So they can function well over a bigger thermal range if you give them enough time to acclimate. We also predict that in general, the wild fish would show, if this hypothesis is correct, the wild fish would show a more shallow slope, and the lab fish would show a steeper slope in biological rates like metabolic rate or enzymatic rates or things like that. All right, so to test this, we used 300 wild zebrafish, 300 lab zebrafish, and they were raised in identical conditions in our lab. So we had a couple of these, we had 3 of these rooms with a lot of aquarius. And then we took them from 28 degrees. These are temperature readings from each aquarium, and we took them up to destination temperatures and down to destination temperatures. So we had one aquarium for each second temperature, from 38 down to 10 degrees. The 38 degree fish started dying after a week, so we terminated that treatment. And then we measured a lot of things. We measured growth, metabolic rates, performances of various sorts, and we did some gene expression and things like that. So I'll show a few of these results. First of all, this is what the fish look like at 30 degrees, which is close to their optimal temperature. They swim around happily. And this is what they look like at 10 degrees. And I have not slowed this down. This is not slow motion. This is real pace. So you can see that the pace of these fish have really slowed down. They are unable to swim normally. If we take them down to 9 degrees, they actually just sink to the bottom and just lay there and don't do anything. So this is really on the limit where they can perform. Some results. So again, wild fish in green, lab fish in blue. We saw that this is resting metabolic rates. We saw that the lab fish seemed to have a steeper reaction norm, which was similar to the prediction. We didn't see that in the statistics. The maximum metabolic rate, however, we saw that the wild fish were able to maintain a broader or decent performance, decent oxygen uptake during maximum swimming over a broader temperature range. So, and then the lab fish in a more narrow range. We thought maybe the lab fish would have a higher peak, but the amplitude was, or the maximum was about the same. But really the wild fish were able to acclimate better to colder temperatures. And this also shows in the aerobic scope, where the wild fish have a broader performance curve. For maximum swim speed, that didn't quite translate into to better maximum swim speed. Slightly higher swim speed in the wild fish, but not much. Reaction time is a fun experiment where we drop a hidden weight down onto the surface, and then we video fish at 1,000 frames per second, and then we just count the time from this weight hitting the surface to the fish doing the first muscle movements, and that is the reaction time. How long does it take before the muscles start contracting? And that is, of course, temperature dependent. We saw that the fish were very fast around optimal temperatures, but then as at colder acclimation temperatures, the lab fish were unable to maintain decent reaction times, and they had actually very, very slow reaction. For acute thermal tolerance, there wasn't much. Acclimation temperature had, first of all, a dramatic effect on thermal tolerance, but there was only a tiny effect of, it was very statistically quite clear, but the effect size was quite small. And again, here, the lab fish have a steeper reaction norm than the wild fish. We also did some gene expression, and I'm only going to show this one. So this is a heat shock protein enzyme gene expression, so HSP70 and HSP90. And this is gene expression after a month at acclimation temperature. So it's not an acute response to warm temperatures. We think that many temperatures had acute responses, but then after a month, they shouldn't need more heat shock proteins unless they are thermally stressed. And here we can see that the lab fish have much higher levels in most of these. So it's muscle, liver, and brain tissue. So in most places, the lab fish have a higher expression of heat shock proteins. And we think this is because they are more thermally stressed. Even though they have been at the destination temperature for a month, they are still experiencing more thermal stress, so they need the heat shock proteins. So again, this suggests that the wild fish are better at acclimating to those high temperatures. For growth rates, we didn't see the expected pattern. This was quite unexpected. We saw that the lab fish actually had higher growth rate, almost twice the speed over the entire range, or growth rate over the entire range. And the wild fish were quite a bit lower there. And this is maybe not so surprising if you consider the fact that they have been, the lab fish have been domesticated for a very long time to work well, function well, grow fast in those conditions, and they probably haven't had much need to for safety or things like that, but instead high growth and high fecundity has been in the lab setting has been successful. So, and also adaptation perhaps to a dry diet. They were fed both flakes and artemia, but it's still a lab diet that they are adapted to. We saw some differences in growth hormone expression and things like that ties into that. Okay, so to conclude this first acclimation experiment, we saw that yes, the wild fish seem to have broader thermal window in many of these experiments. They had a better acclimation capacity is our conclusion there. And the lab fish then have a reduced plasticity, so meaning that potentially plasticity has been costly or has been lost for some reason. It's quite rapid evolution. They have higher growth rates, and possibly this has been a trade-off with plasticity. If they lose plasticity, they might have a higher, be able to reduce that cost of maintaining plasticity so that they can grow higher. But that's that's speculation for now. So performance-wise, we didn't see that very high peak in the lab fish, but maybe they don't really have a big need for performance peak, but they performed about as well as the wild fish in many things, but they didn't have the broad thermal performance curve. All right, so that's the acclimation experiment. Then I'll go over to the second one, the adaptation, which is even cooler, I think, because this is evolution of thermal tolerance. And this was just published in December of last year. So the aim here was to just basically get a rough estimate of how rapid evolution can be. So climate change related, obviously. So starting off with an ancestral population with a frequency distribution of thermal tolerance, if we select on the high performers and the low performers over generations, we can see that we can expect these thermal tolerance distributions to diverge. And then a second aim is to compare these evolved or adapted populations to figure out what kind of mechanisms might be driving this difference. So we started with these wild-caught zebrafish. We used a CT max, critical thermal maxima experiment where we had about 10 fish in these tanks, and then we had a heating coil in heater and a pump that flushed water over that heater so that the arena was well stirred the whole time. And then we had a precision thermometer that measured the temperature, and then we just picked the fish out as soon as they started to lose equilibrium, put them in optimal temperature water again, and recorded each individual's thermal tolerance. And the fish recovered immediately, and they were fine to reproduce later. So we could use the exact score of the parents, the exact thermal tolerance of each individual. So it was quite a big data set. So we did this over, so we started with 1,200 fish. We had a random group that was randomly selected to as a control group. So this was just bottlenecked to the same degree at each generation, but not selected on thermal tolerance. And then we had took the top third of performers with the highest thermal tolerance and made 2 replicate lines, and the lowest third and made 2 replicate lines of that treatment as well. And then we did this for 6 generations with 2,100 fish in each generation. 2 replicates. So each population had 450 fish, and each treatment had twice that many. So in total, this was over 20,000 fish that we measured over 3 years. So this was, lights are going out. So this was quite a big experiment that Rachel Morgan led over her PhD, much of her PhD. So this is the main growth room where we grew the fish, and this is the main room where we did the reproduction and selection. We kept them in larger aquaria, and not the standard small zebrafish tanks, because these are wild zebrafish, and they were happier in larger populations. So what we did was this is a real data. So this is the distribution that we found in the first generation in the F0. And we took the top third, all the ones to the right of that line, to the high line. And these are the down selected. And this is the distribution of the control. Then we also had an acclimated up selected line. So where we gave them 2 weeks at 32 degrees to prepare for warming, and that allows them to induce thermal tolerance, to start their plastic thermal tolerance, to induce genes that promote thermal tolerance. So I'm not going to talk too much about that one because it's a little bit complicated, but these are the results. So here's the thermal tolerance on the y-axis, and on the x-axis is the generation. So 7 generations from 0 to 6. And you can see it's a bit noisy. You can see that they're all going up over generation 4, they're all going down on generation 5, and then up again. So to get rid of that probably environmental noise, it's still quite small. It's 0.3 degrees or so up and down, but still we can get rid of that noise by just centering it on the control lines, the green control lines there. And then we can see that yes, both up lines are increasing in thermal tolerance only by about 1/4 degree. The down lines go faster downwards, so they lose upper thermal tolerance by a much faster rate. Then we can calculate how rapid is adaptation per generation. And the control-centered values here, we see that the increase is 0.04 degrees centigrade per generation, which is quite low. Downwards, it's a bit faster. We also calculated the heritability of thermal tolerance, and up it was of course slower, about 0.1. We calculated this using the Breeder's equation, and down it was more than twice as fast. And these are also centered on the random, on the control line. We found, one of the surprising things was that we found a hard limit to acclimation and adaptation, because we think the closer they get to their upper thermal tolerance, the less room they have to acclimate or adapt. So because what we found in the acclimation group, the red lines here, we saw that the acclimation at the start in the first generation increased thermal tolerance. So a 4-degree acclimation increased thermal tolerance by 1.3 degrees. But then that effect or contribution of acclimation, the effect of acclimation went down with selection. So when the up-selected lines increased in thermal tolerance, they had less potential to acclimate on top of that. So in the last generation, it was less than 1 degree that they could acclimate. So there seems to be a functionally, at least, a trade-off between adaptation and acclimation. So the way we view it now is that there is some form of hard ceiling, which is very difficult for evolution to tackle. And then there's genetic variation in the population that allows adaptation, allows evolution to occur in over generations. So this is the starting generation, and then with selection we increase this up to a higher thermal tolerance. But the acclimated ones, so this is the acclimation temperature down here. The acclimated fish, if you acclimate them to 32 degrees, they are closer to their hard ceiling. So evolution and acclimation has less room to adjust thermal tolerance. So we think that the up-selected, yes, they increased by 0.04 degrees per generation, but at the same time, the acclimation that they can do on top of that adaptation for each generation declined. So yes, they evolved higher thermal tolerance, but their acclimation capacity actually decreased, eating up at least a part of that gained thermal tolerance, which is not good news. All right, so let me conclude this. Yes, we saw evolution of thermal tolerance over a very short time, and that's very cool. It was only 7 generations in total, and we saw very clear evolution, and all lines within treatments diverged in the expected way. So yeah, that was cool. Plasticity didn't evolve in our experiment. Maybe it's difficult for plasticity to evolve. We thought that by having this acclimation group where we allowed them to express thermal tolerance genes, we would evolve plasticity, the response to the acclimation response. We thought that would evolve. So if we look at these results, that would have meant that it would have been the other way, that it would have increased. The response, the contribution of acclimation would have increased for each generation, but that did not occur, unfortunately. So we think there is this hard ceiling that might be due to protein denaturation or membrane fluidity having some limit where cells are lysing, or something similar like that, that would be probably evolvable over very long timescales, but not within the standing genetic variation that populations have right now. This 0.04 degrees per generation, that is actually the rate of global warming right now. So if a fish reproduces every year, it might look like that would be able to mitigate or to adapt to that rate of warming. But this is an extreme selection pressure. We kill off or remove 2/3 of the population, and that's not probably the selection pressure that occurs in nature. It's quite extreme. These fish have a very short generation time of 1 year. In our experiment, we had a generation time of 3 months, but for each generation, it was 0.04 degrees. But most fish species have longer generation times, and if we look at Greenland sharks, for example, they mature, they become reproductive at 150 years old. So yeah, evolution is very slow in some species. And then we have this trade-off with plasticity. As they adapted higher and higher thermal tolerance, the ability to acclimate decreased as they approached this hard ceiling. Yeah, so these selected lines, we have them and we will continue working on them. We're looking at which traits are correlated to this. What are the mechanisms that have evolved? Is it oxygen limitation, circulation, brain function? We're still investigating that. And also the genetics, which genes were mostly evolved and how do they relate to physiology, the phenotype that we see. So those are things that will keep us busy for a couple of years more. All right, then I just want to acknowledge all the people who did the excellent work, my group and everyone, all the students who helped with the selection and the funders. All right, thank you.
Thank you so much for that great talk and for joining us today. I'd invite anybody who has any questions. You can go ahead and unmute and put your video on and ask whatever questions you might have.
So I have a question. First of all, that's a very nice presentation. Thank you very much for your presentation. You did mention membranes, and I thought this is very interesting that as temperatures warm, you're going to want to put more cholesterol in your membrane, reduce the amount of polyunsaturated fatty acids in the membrane. Yep. And so there is a dietary component. And what's quite interesting about sort of the ecological studies, of course, is that the entire food web will be responding to an increase in temperature. And so you can expect the diets to change naturally with an increase in temperature to produce less fluidity through lower PUFA and more cholesterol. I wondered if you thought about maybe doing some dietary experiments as well.
Yeah, that's an excellent suggestion, actually. Rachel, who was the first author of this study, is doing a postdoc with Neil Metcalf, and they are actually doing exactly that, manipulating diets and the fatty acid profiles of the diets to see if that affects thermal tolerance. We haven't done much on it here. We did a gas chromatography sampling from the first experiment, the acclimation experiment, but the samples weren't good enough to draw any conclusions. It was just noise, unfortunately. We haven't done it yet on the adapted lines, but that's something that could be done. And I mean, we know that organisms can adapt their membranes to tolerate extremely high temperatures, but it might require longer times than what we have here, and what we might see with climate change as well.
And another one to watch out for is cholesterol as well. So yep. Cholesterol has a condensing effect. And the work we've done suggests cholesterol is actually quite an important, at least in trout, when you start to raise, anywhere close to their temperature toleration, we see a very dramatic change in cholesterol. So it would be something else to look at too.
Yeah, we just did fatty acid profiles. We don't think we can see cholesterol on that.
Something for the future.
Yeah, all right, we'll do that.
Jump in with a question if that's cool.
Yeah.
Cool. Thanks for the talk. That was great. I had a question. What do you think the trade-off would be if you took your F6 fish that were up-selected for a higher CT max and then you ran your experiment in the other direction? Is there going to be a trade-off for their ability? Are you going to narrow their window too much at these high acclimation temperatures to be able for them to acclimate back in the other direction?
Yeah, that's a good question. I mean, for if we want to do that over multiple generations, I think there is a little bit of bottlenecking. So if you're thinking of adapting them over and selecting them in the opposite direction over several generations, I think genetic variation in the populations is reduced over time in these because we have a generation size of 150 individuals in each of these 8 lines. Yeah. But there is not all of them reproduce each generation. So there probably is a loss of genetic variation in the lines. So I think responses to selection will probably go slower if you start doing it the opposite direction. Acclimation capacity from the first experiment, we saw that that's maintained quite well. So even though our predictions were kind of clear in many of the measurements, with the loss of acclimation capacity in the lab lines, we still saw that they weren't bad at acclimating. They were still quite good. It's just that the wild fish are extremely good at acclimating. So, and that was 150 generations and probably a bit of inbreeding and loss of plasticity. So acclimation capacity seems to be maintained, and I would imagine it would be maintained over this selection experiment as well.
Cool, thanks.
Yeah, thanks.
Kurt, I saw that you had a question on Facebook. But you just joined. Do you want to go ahead and ask, or would you prefer that I read it?
Hey, Frederick, it's
a question on Facebook. You just joined. Do you want to go ahead and ask?
Yeah, sorry, the link didn't work for me originally. It asked for a password, so I had to join in through Facebook and watch the talk. Thanks for the talk, Frederick. It was really interesting. So obviously these are really, although they're on the high end of temperature for fish, they're quite eurythermal. So what do you think, for example, that fish have a much narrower normal thermal niche? Originally it asked for a password, so I had to join. Whoa, what's going on? Thanks for the talk, Frederick. It was really interesting.
The Facebook feed is a little slower than the Zoom call, so if you're listening through Facebook, it'll be delayed.
So Can you hear me now?
Yeah.
Okay, I turned off the Facebook. I think I had them both on. I guess the question is, if you have an animal that's normal environment isn't as variable in temperature, what do you think that where they would be relative to that 0.04 degree per generation? Do you think that would have more potential for evolutionary gain or less?
Yeah, it's a great question. I think if they are very close to their upper, or if they're tropical fish, I think they might have less ability to adapt, because they are going to be, if you look at some coral reef fish or something, they might be very close to their upper limits, their long-term limits. And that probably means both lower acclimation potential and lower adaptation potential. But if you're looking at a temperate fish that is not as euthermal, maybe they have a faster adaptation response. But yeah, it's hard to tell. I think temperate fish, I'm more hopeful for temperate fish that they have a high response. But the problem is their generation time is so slow, so it doesn't matter.
No. Yeah. Yeah.
So
there'll be a real disadvantage.
Sorry, what's that?
I said they'll be at a real disadvantage.
Yeah, exactly. Yeah. Yeah, if your generation time is 10 years, it doesn't matter if you have a good response to selection or to, it's going to take too long anyway.
Hi, Frederick. Are you okay if I went ahead and asked a question as well?
Yeah, of course, of course.
Okay. So the experiment in the last experiment that you were talking about with the zebrafish, you've been testing the CT max in adult fish, but I was just wondering, what do you think about other life stages of the zebrafish, like the early life stages, for example, when we were talking about eggs and embryos and larvae? I've been reading that at these life stages, species can actually be a lot more vulnerable to warming than
There's
adults.
a lot of noise. I have a hard time hearing you.
Okay, I'm not sure why. Is it better right now?
Yeah, it's fine. I can hear you fine. Yeah.
I'm so sorry.
The Jet Propulsion Laboratory in Southern California.
Oh, it's the Mars landing starting.
We're gearing up for NASA's Perseverance rover to touch down on Mars. Happening in an hour and a half from now.
Okay, we have an hour and a half. That's good.
Land in Jezero crater. The most difficult landing ever attempted. Thank you for joining us. I'm your host, Raquel Villanueva. In the past, mission team members gathered in our mission control for landings.
Okay,
I'm gonna. Around, we have COVID safety measures in place. Today's landing will look a little different than what you've seen before. I am in a room by myself.
Okay, I figured out what the problem was. It was me. I had the sound on. It was a live stream starting. Sorry about that.
No worries.
Okay. All right. Let's. Can you repeat the question? I got distracted by the NASA live stream. Sorry about that.
No problem at all. Okay, so yeah, I was just wondering what you thought about earlier life stages of the zebrafish, and in terms of their vulnerability to warming, because I've been reading some work by others emphasizing that the eggs and the embryos and the larvae of fish might actually be the bottleneck in terms of how they're going to respond to warming in their environment. At this life stage, fish might actually be more vulnerable than adults. And your experiment has been done with, you've been testing CT max in adults. So yeah, I was just wondering what you thought about that.
Yeah, that's an excellent question. And this is the Fleming-Dalke paper, for example, did a big study on that. And concluded that, yeah, early life stages are much more vulnerable. I think that is probably true for cod and for the species that they have investigated. It's not true for example, zebrafish that reproduce in the cold season, where everything is flooded and there's lots of cold water around. So I think it's very dependent on the life history of the species, and some animals or some fish will spawn in colder, deeper water or cold lakes or whatever, and others will have broadcast spawning that float to the surface, and they might be very vulnerable. So I think it's very dependent on the species. So I think it's wrong to generalize like that. So I think for zebrafish, we know that the water in the wild reaches 39 degrees. And we saw in our lab experiment that they can tolerate 38 degrees for 1 week, and then they start dying. So the conclusion is, yes, they are dying from heat stress in the wild already. And with a 4-degree projected increase in water temperature in India, or in freshwater temperatures in India, most of the, I don't know, a lot of the freshwater streams, the shallow freshwater streams, are going to be too hot for a lot of fish species. And not during the wet season when everything is flooded, because then the temperatures are cold, but during the dry season when the water is stagnant and shallow.
Yeah, that makes sense. Yeah, I think I also read a study about larvae in Atlantic or in Pacific salmon, I think so. And I guess that in these kind of species, the early life stages are maybe the most sensitive in terms of heat exposure, because the fish are laying their eggs in freshwater streams, and that's where temperatures are the highest. But yeah, you're completely right about the zebrafish being in a different environment. And
but in some species, it might even be the adults, the spawning adults that are the vulnerable life stage, if they are migrating up rivers or the temperatures get too hot to supply oxygen to big gonads and things like that. So yeah, I think it's too early to generalize and say it's always early life stages. But yeah, probably in a lot of species, yes.
Yeah, but maybe not in zebrafish. Okay.
No, probably not in freshwater fish in India.
Yeah, okay. Thanks so much.
Yeah, thanks.
Are there any more questions? Give it a minute for anybody who's shy. Looks like no, so I think we'll end a little bit earlier than we anticipated, but thank you so much for coming to speak to us today. I really enjoyed the talk, and I'm glad we were able to have some good discussion.
Yeah, thank you so much for inviting me. It was my pleasure to talk with you guys. And I mean, I see a lot of names here that I recognize, and I read the papers coming out of your lab, and it's good to see you guys.
Have a nice day. Thank you.
All right. See ya.
Have a nice evening over there.